COVID-19, commonly known as coronavirus, has severely impacted all of our lives. It has changed the way the world has functioned at a society whole and on an individual level. The virus is made up of a lipid protective bilayer that is home to three types of proteins: the spike protein S, the envelope protein E, and the membrane protein M. The S spike protein is unique as it has the ability to bond to ACE2 receptors. This bond allows for the transmission of the virus's genetic material into
Since the outbreak of the novel coronavirus COVID-19, the world has been desperately searching for answers. These images are the results of a study that evaluated the effectiveness of 5 FDA-approved antiviral drugs that already exist. Favipiravir is not FDA-approved, and remdesivir has only recently been approved under an emergency basis. These graphs assess the drug's ability to fight COVID-19 by measuring its ability to inhibit the virus from being able to infect and the ability to slow or stop viral infection once infected. The percent of cytotoxicity in these graphs refer to the drug cytotoxicity, which offers us some insight into the safety of these drugs for human cells throughout the body. The most promising drugs based on this graphical analysis are chloroquine and remdesivir, which is why we decided to focus our project on these drugs and include hydroxychloroquine because it is widely considered to be a safer version of chloroquine. Both chloroquine and hydroxychloroquine are weak bases and have the same 4-aminoquinoline core structure. However, they differ in their basic side chains. Their weak basic nature has significance in the drug's mechanism of inhibition to fight against the virus because their basic side chains allow them to accumulate in endosomes, altering the pH towards basicity, which prevents the virus from fusing to human cells. Both drugs have 3 functional groups with different pKa values, and due to the hydroxyl group present in hydroxychloroquine, chloroquine is slightly more basic. Studies found that eye conformations of hydroxychloroquine exist at higher concentration in the blood, which may suggest that the effectiveness of the drug and resulting side effects are stereoselective. Chloroquine, on the other hand, was found to have virtually similar effects on the body regardless of stereoisomerism. For full time in this graphical comparison, each drug was added to the cells an hour prior to being introduced to the virus. The cells were then kept in a drug-containing medium until the end of the experiment. For entry, these drugs were added to cells an hour prior to viral addition and re-added 2 hours post-infection. However, they received no further drug exposure for the rest of the experiment. The post-entry column, the drugs were not added until 2 hours post-infection, but were maintained throughout the rest of the experiment. We interpreted that this data shows that chloroquine may be the more effective drug. However, the ability to inhibit the virus once the patient has already been infected seems to drop nearly 50%. You might be thinking, well, it's inhibiting at least 40%, right? So why wouldn't this be used? Or why can't we all just get prescription for this until a vaccine comes out? This begs for us to consider the side effects such as heart damage, abnormal heart rhythms, vision problems, low blood count, and rash, as well as the costs and implications for the synthesis of this drug. While there have been studies of a similar drug, hydroxychloroquine, considered to be safer, prolonged usage of this drug as well leads to similar side effects. Side effects for remdesivir still remain unknown and very uncertain as it's still going through clinical trials.
So this method to synthesize the key intermediate for creating hydroxychloroquine with the oxygen hat is called batch synthesis, which is basically like when compound A turns into B and then B turns into C, et cetera, kind of like what we've been used to seeing in lab and when we make organic compounds in class. And this traditional way has worked for a really long time, like we've been using it since the 1950s to make chloroquine, but the reaction can usually take anywhere between 24 to 48 hours just to make the intermediate for hydroxychloroquine. So with COVID-19, now there's a shortage of this drug, and people with lupus and rheumatoid arthritis need it. And if there's a way to make this better tolerable for people who've been diagnosed with COVID-19, we're definitely going to need a quicker and better synthesis of the drug. So there is an alternate way of doing this by using a transition metal catalyst, and this way we can just get an SN2 substitution from the original starting material, which sounds great, but this process actually took 18 hours and the yield was only 35% when researchers actually did this, which isn't great either just for the intermediate, for the end goal of hydroxychloroquine. But in a study done by you and colleagues in 2018, they actually showed a new method of synthesizing this key intermediate using flow synthesis. And it actually only took 6 hours to yield 75 to 80% of the product. And with flow chemistry, you can have multiple inputs that meet at the same junction at the same time, which allows for a faster reaction. So here is the proposed retrosynthesis of a hydroxychloroquine, and I was just excited to show a retrosynthesis in this way, and when I saw it in the study, since we're so used to seeing it in class. Here we're looking at the first two reagents that are added to the reaction vessel to yield this iodine keto intermediate, and the reaction time was actually only 5 minutes for all 6 trials. So we're off to a good start. And in order to create the SN2 reaction with the amine group, we took the previous piece and put it in the reactor with this amine and created this oxime piece, which is just the double bonded nitrogen with the hydroxyl group. And as you can see, this process only took 10 minutes for all the trials. So again, it's still looking like a pretty good synthesis for this key intermediate. And then finally, they hydrogenated the oxime by adding dihydrogen to the reaction vessel, which also contains Raney nickel catalyst, and this part only took 4 hours to complete. So now we have the key intermediate, but to get the job done, we have one more step, and it's to combine 4,7-dichloroquinoline with this newly synthesized key intermediate. And in order to do that, the researchers used potassium carbonate and ethanol to create a weak base that would perform the substitution and finally make hydroxychloroquine. This reaction actually took 6 hours, just this piece, but it's still much faster than taking 48 hours just to make the intermediate. Also, the yield for this reaction was around 75 to 80%, which seems to me like this is the faster and more efficient way to make the drug for people that need it.
The first reaction in the synthesis involves an acid-catalyzed hydrolysis of the ester, followed by a decarboxylation, followed by an SN2 substitution. Hydrochloric acid and water creates H3O+, which along with heat leads to an acid-catalyzed hydrolysis of the ester. First, H3O+ protonates the carbonyl oxygen, resulting in a resonance-stabilized intermediate with a positive charge that can be delocalized onto carbon or either oxygen. Water then adds as a nucleophile to the carbon, and then a proton can be transferred from the H2O group to the oxygen in the ring. The ring then opens, resulting in another resonance-stabilized intermediate with a positive charge that can be delocalized onto oxygen or carbon. The oxygen double bonded to the ring is then deprotonated, which reestablishes the carbonyl group. The dicarbonyl compound then undergoes decarboxylation, releasing carbon dioxide, similar to an acetoacetic ester synthesis or a malonic acid synthesis. Hydrochloric acid then protonates the alcohol group, creating an H2O group, which is a better leaving group. The chloride anion then attacks as a nucleophile and displaces the H2O leaving group. Attacks as a nucleophile and displaces the H2O leaving group. The second step in the synthesis is protection of the ketone. Ethane 1,2-diol and tosylic acid are used to convert the carbonyl into an acetyl. Protecting the ketone prevents nucleophilic addition of the amine reagent used in the following step to the carbonyl carbon. The third step is an SN2 substitution occurring after addition of 2-ethylaminoethanol, toluene, and heat. 2-ethylaminoethanol, which is a secondary amine, attacks as a nucleophile and displaces the chloride leaving group. The fourth step is deprotection of the ketone. Hydrochloric acid in water creates H3O+, which along with heat is used to convert the acetyl back to a carbonyl. A proton then transfers from the ammonia nitrogen to the oxygen, creating an alcohol group. Next, the pH is adjusted in order to protonate the alcohol group, creating an H2O group, which is a better leaving group. The lone pair electrons on the nitrogen then establish a double bond to the carbon and H2O departs as a leaving group. The H2O deprotonates the nitrogen, creating an imine. Next, the imine is reduced to its corresponding primary amine. This reduction is done by catalytic hydrogenation using Raney nickel, which is a metal catalyst from nickel-aluminum alloy that had most of the aluminum removed and contains hydrogen gas on its surface. Catalytic hydrogenation can be used because there are no functional groups that can be reduced other than the imine. Therefore, a selective reduction of the imine to an amine using NaBH3 and ethanol is not necessary. The sixth step of the synthesis is a nucleophilic aromatic substitution with an addition-elimination mechanism occurring after addition of 4,7-dichloroquinoline and heat. The nitrogen para to the chlorine in the 4,7-dichloroquinoline ring is the equivalent of having a strong electron withdrawing group para to the leaving group. Therefore, an addition-elimination mechanism can occur. The primary amine created in the previous step acts as a nucleophile and adds to the position para to the nitrogen. This results in a resonance-stabilized intermediate with a negative charge that can be delocalized through resonance onto the nitrogen as well as other positions on both rings. Chloride then departs as a leaving group and aromaticity is reestablished. Finally, the amine nitrogen is deprotonated, yielding hydroxychloroquine.
The global pandemic of SARS-CoV-2 has driven the biomedical community to develop antiviral interventions. One potential drug currently being evaluated in numerous clinical trials is remdesivir. Most of the world had not heard of remdesivir until a few months ago. It's not a brand new medication. It was developed by Gilead Sciences during the West Africa Ebola epidemic, and they began testing remdesivir against SARS-CoV-2 in January. They recently got FDA emergency use authorization because remdesivir demonstrated efficacy in both in vitro and in vivo models against coronaviruses. Another research published by University of Alberta shows remdesivir to be highly effective in stopping the replication mechanisms of SARS-CoV-2. Remdesivir is a nucleoside analog structurally similar to the nucleosides that make up DNA and RNA and is specifically an adenosine analog. Replication of SARS-CoV-2 requires viral RNA-dependent RNA polymerase. The active metabolite of remdesivir interferes with the action of RNA-dependent RNA polymerase and tricks it into incorporating remdesivir in the genetic code of the virus instead of adenosine. Let's take a look at how the active metabolite is formed. Upon diffusion of remdesivir into the cell, the reaction is initiated by esterase-mediated hydrolysis, giving carboxylate that displaces the phenoxide. The unstable cyclic anhydride formed is hydrolyzed by water to the alanine metabolite. Alanine metabolite's phosphorus-nitrogen bond is hydrolyzed by phosphoramidase enzymes to liberate nucleoside analog. Further phosphorylation yields the active nucleoside triphosphate that inhibits viral replication. The Cairo electron micrograph reveals RNA template inserted into RNA-dependent RNA polymerase, where remdesivir is covalently incorporated into the primer strand. The synthesis of remdesivir is done by first preparing the intermediate A from mellalanine and phenylphosphodichloridate in the presence of triethylamine and dichloromethane. Then ribose is protected by triple benzyl and the molecule is oxidized by dimethyl sulfoxide with acetic anhydride that gives the electron intermediate B. 4-aminopyrrolotriazine is brominated and the amine group is protected by excess trimethyl chloride. N-butyllithium undergoes a bromide-lithium exchange reaction at low temperature to yield the intermediate C. The intermediate B is then added to a solution of intermediate C. This reaction is carried out in a weakly acidic solution to obtain anomer mixture. The mixture is then reacted with an excess of trimethylsilyl cyanide in dichloromethane at low temperature for 10 minutes. It is then quenched in an aqueous solution of sodium hydrogen carbonate to obtain a nitrile intermediate. In the next step, benzyl protection is removed with boron trichloride in dichloromethane at low temperature. The isomers formed are separated by reversed phase high-performance liquid chromatography and reacted with trimethyl phosphate and methylimidazole to obtain a diastereomer mixture of remdesivir. In the end, using chiral resolution method, optically active remdesivir in its pure form is obtained.
Soap is derived from triglycerides, which are typically found in animal fats and vegetable oils. Triglycerides consist of a glycerol backbone and a fatty acid tail that is held together with a relatively weak ester bond. During saponification, the triglyceride bond is first treated with a strong base like sodium hydroxide. The second step in saponification is an irreversible step that involves the cleavage of an ester bond and the formation of a glycerol and fatty acid salt. Soap contains Each soap molecule contains both a hydrophobic tail and a hydrophilic head. The hydrophobic tail is nonpolar in nature, made up of mostly carbon chains, and is non-soluble in water, whereas the hydrophilic head is polar, ionic in nature, and soluble in water. It is due to this opposition in charges and separation that typical soaps and detergents form micelles. The hydrophobic tail of the soap molecule is attracted to the lipid bilayer of the virus. This attraction creates a jackhammer-like force that begins to break through the protective lipid layer. Once this is broken and compromised, the S protein spikes that typically carry RNA are unable to transfer the genetic material into the human cell. The recommended time to wash your hands effectively is approximately 20 seconds, which is also like singing happy birthday or reciting the ABCs twice, or like singing Britney Spears' Oops, I Did It Again chorus twice. So in conclusion, just wash your hands, please.